been developing a lot of nanocarriers in the labs, but we are unable to translate this technology into the market or in the clinic. Why are we unable to translate this technology? What are the bottlenecks? So there are some manufacturing challenges like scalability. Like I have told you, the solvent evaporation method. We can, when we are making a small batch, like in the lab, we can do solvent evaporation, but we can't use this method when we to make a dose or to make a batch that will be used, that will be made for whole in the whole market. Means a market size batch can't be made using this what do we call syringe and the beaker. We can't means scale up this process, and there are a lot of batch to batch variability. Why? Because nanoparticles, they are very sensitive to minute changes in the process, manufacturing process. Like if the solvent quality is also like changed, or it varies from batch to batch. If we don't take the solvent from the same manufacturer, the minute change in the solvent quantity, it will change the target profile, means the profile of the nanocarriers, it will be changed by minute changes. So these processes, the development processes, these are very critical, and these are very sensitive to these small changes like rpm also, the rate at which you are stirring your sample. If we change it by just 50 or 100 rpm, the whole product will change. So these variability, minute variability, they these are one of the bottleneck. By these minute changes, we can't tolerate these manufacturing, means the manufacturing of nanocarrier. We can't tolerate even minute changes in the manufacturing process. And cost of production, like when we make a tablet, the excipient which are used are not very costly. These are economic, means they are very cheap. But the lipids and polymers which are used for the development of nanocarriers, these are very costly. Like PLGA nanocarrier, PLGA polymer, which we use to develop PLGA nanoparticle, the cost of 50 mg of PLGA polymer is around 50,000. So it is very costly, and the everybody can't. Just a minute. And everybody can't afford this. Everybody can't afford costly medication. So we need to see, like in the developing nation like India, if we are making a nano carrier using such costly materials, not all the people will be able to afford that. And there are also some safety and efficacy concerns, like toxicity concerns. Like nanocarriers, they are made up of synthetic agents, and when we and these synthetic agents, like if we put any synthetic agent inside our body or inside our blood vessels, body will and uh, try and eliminate it, because body won't let anything stay inside it if it is not endogenous, means it's of self nature. Body will consider it as a threat and will try to eliminate it or remove it. So, and narrow carriers, they can also cause immunogenicity. Immunogenicity means they can trigger some immune response in the body, which can lead to allergic reactions and anti-medicine antibodies. If anti-medicine antibodies are generated, then the whole of the medicine will be eliminated out of the body, and we will not be able to see any effect or any therapeutic effect. There is off-target accumulation also, because when we give nanocarriers via intravascular route, the macrophages which are present in the spleen and liver, they try to capture these nanocarriers, and thus when we when these nanocarriers, they are captured by the macrophages present in the liver and spleen, they will accumulate over there, and they can show some side effects, and they will not reach at their specified site, the desired site, and they will reach liver and spleen, and they will show some off target accumulation and off target effect. Lack of targetability also. So nanoparticles, they are unable to extravasate. Extravasate means when we inject a nanocarrier inside a vessel, it needs to come out of a vessel to show its activity. If it won't come out of its vessel, uh, it can't show the specificity, means it can't target that particular cell. So it needs to come out of the vessel to show its activity. But nanocarriers, they don't have any such property by which they can extravasate out of the vessels, blood vessels. So we also need to work on this aspect of the nanocarriers such that we can improve the site specificity or targetability of the nanocarrier. Lack of predictability also, as I have told you, a, sa- a small, minute change in pH and temperature can affect the performance. Uh, like in a tumor, there is a certain change in pH and temperature. So this pH and temperature can also affect the performance of the nanocarrier. Entrapment of macrophages, I have already told you. These macrophages, they try to eliminate the nanocarriers, and if the nanocarriers are eliminated, we are unable to see any activity. So here I will try to tell you regarding the application of nanocarrier in the cancer or tumor drug delivery system. So anti-cancer therapy. So most of the anti-cancer drugs, they are hydrophobic in nature, and so if